Welcome to part 2 of protein synthesis in year 3 of science. And in this lesson we are going to continue our discussion of how proteins are synthesized in the cell and what do they make. Okay, so here we have just an illustration of a few examples of the thousands and thousands of different proteins that the cell can make. And they really are amazing machines, are very beautiful really in their structure. So we are going to see how this happens. So in the last lesson we were talking on how the messenger RNA enters the ribosome and the three codon of nucleotides attaches to the transfer RNA that matches that codon with the anticodon, and it adds into the ribosome one more amino acid to a growing chain of amino acids. So this chain is what is going to form the protein. So here we have another representation of the same thing. This is going in the other direction here in the picture. But you see, I like this picture because it shows the ribosome in less detail, but it shows now the messenger RNA and how the three nucleotides are matched. And then the transfer RNA comes in here, and you see how it matches the codon and the anticodon. So they join together. AAA is matched to UUU. GAU is matched to CUA. Okay. And then the next one, which is UUC, will be matched to this transfer RNA, AAG. Each one of these different types of anticodons and the transfer RNA that has that anticodon attracts one specific type of amino acid. And so then the amino acid is joined into this growing peptide chain, which is going to form the protein. And then the uncharged transfer RNA is released back into the cytosol in order to go and find another amino acid of that same type. Okay. So the amino acids, we talk about how they are composed, and they are these 22 are the ones that are matched in the ribosome, and they have specific names and a specific molecular structure and different characteristics, a different geometry, electric charge. Some are positively charged, some are negatively charged. Okay, some have rings that form a more bulky structure. Some are hydrophilic, so they are attracted to water. They are water soluble. Others are hydrophobic. They are like lipids, more like lipids, so they repel water. So here we have this group are the hydrophobic side chain. You can see they don't have much oxygen in the side chain. These are the ones that have an electrical charge. So these are the basic amino acids, and many times they are referred to them by their name or the three-letter abbreviation or just a one-letter code that is given to each one of the amino acids. So when the amino acids are joined together, we mentioned that basically the carboxylic part of one amino acid is joined to the amine side of the other one, and then this oxygen and these hydrogens are joined together, okay, in a water molecule, and the nitrogen is joined to the carbon, and so here it forms a peptide bond, a very strong bond that is going to form the protein chain. The residue here is the part of this part, the tail, if you wish, is many times just denoted as R, or the residue. But that could be a small, could be large, could be electrically charged or with a circle and so on. But that is how the peptide chain keeps building up. So now we have that as this chain, this peptide chain starts exiting the ribosome, is going to start folding, and we have different structures. So the primary structure is what is basically just the sequence of amino acids that form the chain. The secondary structure is basically given when, depending on the particular sequence of amino acids, these amino acids are going to fold or form helixes or twist and turns or chains and so on, depending on the electrical charge, whether they repel or they attract each other, the geometry, the specific structure of the sequence of amino acids that go together. So that's the secondary structure. The tertiary structure is when the whole chain is complete and is basically completely folded in itself. The chain now forms a specific three-dimensional structure. Okay. The quaternary structure is when the three-dimensional structure of one chain of amino acids is joined to other chains of amino acids. It could be other chains of the same type, or it could be that sometimes chains of amino acids are joined to carbohydrates or lipids, or actually strands of RNA, and they form very, very complex shapes, and that is the quaternary structure. And that is actually the protein molecule that is a biological machine that does something, is built for a specific function. Okay, so when the when the protein is going to be used inside the cell, all right, so for example in the cytoplasm of the cell, there's the cytoskeleton. The cytoskeleton is made out of microtubules and filaments and so on. All those things are made out of proteins that are made inside the cell. So if the protein is going to be used inside the cell, the ribosome releases the protein directly into the cytosol, okay? And then the cell would use the protein. If the protein is going to use outside of the cell for something else, then the ribosome actually attaches itself to the endoplasmic reticulum, the ER. And the part of the ER that is studded with ribosomes is called the rough endoplasmic reticulum. Okay, so these purple balls, number 5, okay, are the ribosomes that are attached to the endoplasmic reticulum. And then when that happens, when a ribosome forms a protein and is attached to the endoplasmic reticulum, the protein actually is released inside the membrane of the endoplasmic reticulum. The endoplasmic reticulum is a network with folds and everything in order to increase the surface, but it's a large network of membrane that is attached to the nuclear membrane. So it's always very close to the nucleus. Now, The proteins that are released inside the endoplasmic reticulum are processed further. Okay, so all the tertiary and quaternary structure of proteins, my understanding is that most of that happens inside the endoplasmic reticulum. And so as the protein moves inside the endoplasmic reticulum, it attaches itself to other proteins, it is changed, it's refolded and everything in order to form its proper shape. Then, after that, the protein is basically, when it reaches the end of the endoplasmic reticulum, the protein is carried in a vesicle to the Golgi apparatus, okay? And the Golgi apparatus does more further processing of the proteins. So you see all the proteins kind of moving through the folds of the Golgi apparatus, and then they are transported through vesicles into to the cell membrane or to the outside of the cell, okay? So, so here we have another representation, basically, of the same thing. We have the ribosomes into the endoplasmic reticulum, and then how the proteins move through vesicles into the Golgi network, okay, Golgi apparatus, and then the vesicles take the proteins out, and through the process of exocytosis, then the proteins are released outside of the cell membrane into the bloodstream or the extracellular space, okay? And those proteins then perform their functions, okay? So for example, a cell could produce hormones, okay? And hormones are proteins that are used for signaling. And so once they are released into the outside of the cell, the hormones are released out there, and they send signals to other cells to perform other functions, okay? Sometimes after proteins perform their functions and they are all done with them, or other things, carbohydrates or lipids or other parts of the cell that are no longer needed, they are taken into lysosomes. Lysosomes are like vesicles, they are membrane-bound organelles, but the difference in the lysosomes is that they have a very acidic environment, okay, and they are able to and have enzymes that are proteins also that accelerate chemical reactions, and these enzymes break down whatever is inside the lysosome. So if the lysosome contains proteins, then there are enzymes that basically unfold the protein and start breaking the peptide bonds of the protein, and then they release the amino acids, the parts of the amino acids, back into the cytoplasm to be reused in order to rebuild other proteins, okay? So that is what the function of the endoplasmic reticulum, the Golgi apparatus, the vesicles, and the lysosomes are. And so here we see how not only how the proteins are made, but also how they are unmade and recycled. So we saw this also in the previous lesson. This is just a very, very small example of some of the amazing structures that proteins can build. And proteins are really used for lots of different things, okay? I'm amazed, for example, at this one here. It says insect flight muscle. And see how interesting, how complex it is. It has the different colors identify different protein subunits. Remember when we were talking about this is the quaternary structure of the whole protein, but is composed of different parts and pieces that are attached together, and then they make the whole thing an insect flying muscle. Okay, and so really, some are very very small, others are very large. This is a adenovirus. So a virus is usually a protein that encapsulates inside some RNA, the viral RNA or DNA. Okay, and the protein is just a capsule that surrounds that molecule inside that transmit the disease of the virus. So, but we see that there are amazing machines that can be built here with proteins. And the interesting thing is that depending on the genes that a cell expresses, okay, so depending on which genes, the cell has the whole complete library of the organism, okay, but a cell particularly does not express all the genes that are in the chromosomes. So they only produce certain genes, and they only, because they do certain genes, they only produce certain hormones. Okay, the production of a specific hormones in a cell differentiate that cell and make that cell to be of a specific type. Okay, and that is how we have the cells themselves become very different. They all have nucleus and lysosomes and cytoplasm, the cytoskeleton and so on, but the shape and the proteins that are inside the cell determine what is the cell function. So, for example, nerve cells have a specific shape with dendrites and an axon, a very long axon that communicates to other nerve cells, and they have a strong cellular membrane with a specific signaling mechanisms to transport the electrochemical reaction that makes for the nerve signal. All right, so they're very specific type of cells. The muscle cells also are very different. They have the proteins inside them that make the tissue, the muscle tissue, that can contract and release, and some are, the striated muscle cells are for voluntary movement, that is the movement that we control, and the smooth muscle cells are for involuntary movement. So, like, for example, the movement of our lungs, all right, or our eyes, that we don't control when we kind of control when we close our eyes and open, but many times that happens involuntarily, just because we need to do that. Cardiac cells are also different from those, very, very strong muscles. Bone cells produce a lot of enzymes and proteins that are actually then used to create the bone structure. And each one of the cells is just an amazing, amazing thing. I'm going to go now here to an article in Wikipedia. This one has the list of distinct cell types in the adult human body. Okay, and just see the huge list of different types of cells, okay, that are in the human body. Some are for producing, for secreting different type of glands, like sweat, or the stomach, the gastric glands, okay, and so on. Each one has a specific job and a specific type of proteins and enzymes that they release in order to perform their function. All right. Then we have these ones I think are very interesting. Keratinizing epithelial cells. So epithelial cells are the ones in the skin. All right, and there are some that produce fingernails and hair, and the hair on the head, and the hair on the skin, and so on. And each one is different. We have also the nervous system cells and the sensory transducer cells, the ones for our sensory systems, auditory cells, the eye cells, photoreceptor cells, and so on. An amazing variety of these different types of cells. Isn't it amazing, really? And so here I just click on two of them. The parietal cells, okay, these are the cells that are in the stomach, in the gastric glands, and they actually produce hydrochloric acid. Hydrochloric acid is very strong, very strong. And it's interesting, just reading here, I found that they have a specific enzyme. Remember, an enzyme is a protein that accelerates a chemical reaction. And you can always recognize the enzymes because their name ends with AS. Okay, ASE. All right, so whenever you say an enzyme of that type, you would say, okay, that's an enzyme. All right, this case is called hydrogen potassium ATPase. Okay, so this enzyme is interesting because it's able to concentrate hydrogen ions in a very high concentration of 3 to 1. So it basically makes one side of the membrane, okay, of the cell a lot more acidic than the other, and that is one of the ways that it can produce the hydrochloric acid that then it releases into the stomach. And the hydrochloric acid is basically used to digest things and break down the food that we eat. Okay, amazing cells. Here I click on the photoreceptor cells. Of course, this I was very curious. How these are the cells that are a type of neuron cell found in the retina and is capable of phototransduction, so communicates light, information. Okay, and here I found there are two types of photoreceptor cells in the eyes. One are called cones. Okay, these are the cones, and they have, and see the shape of this cell. So this is the cone part that is in the retina. Okay, and then here you have kind of the cell part, and it has its mitochondria and the cytosol, and then it goes through the kind of a membrane of the retina. The nucleus is on the other side of that membrane, and then there's a foot of the cone cell that is a synaptic body. So this synaptic body joins basically with another neuron cell that transmits the photo information, okay, that is perceived here. All right, my understanding is that the cones are the ones that are able to see color, and the cones, see that they have also the same type of thing you have, but instead of a rod shape, it has a cone. And the cones, if I remember correctly, are the ones that are able to differentiate between black and white, the basic shades, okay, of gray and light and so on. All right, but see the shape of that, and then here it also has that synaptic terminal that joins to another nervous cell that transmits the information of this cell to the brain. Okay. So in this assignment, in the assignment for this lesson, one of the assignments that you can choose to do is to study in depth one of these type of cells, okay? Or the other assignment would be to choose two and do a comparison between the two type of cells and what makes them different. What are the proteins, the genes that are expressed, and the proteins that are expressed in order to make this differentiation. So an amazing, really, the variety of things. Okay, so the principle here that we see is really that each one of us, okay, like the different parts of the cell or the different types of cells, as a person, we are important and we can make a contribution in our life, in our society, and we don't have to wait. Oh, I'm going to make a contribution when I go on a mission, okay, or later on. You can make contributions to your environment right now. I believe that very, very strongly. And just look for the opportunities of what is it that you can do just now. I hope that you enjoy this lesson, and as much as I have enjoyed preparing it for you. Okay, thank you.